When I was doing paragliding, I crashed and I had an accident. I had field surgery. I had lung injury. And I woke up in a helicopter getting a blood transfusion that saved my life. And I remember asking him, was this blood tested? And he said, yeah, it's okay. Don't worry. And as I woke up again, it was already in the hospital. That sparkled an interest in the challenges of blood transfusion. And I understood that I have to find a solution for that. Many years later, I came across a technology originating from the Technion here in Israel that allows to grow stem cells, iPS, at large scale and allows to produce these cells at a very low cost and differentiate them into your target.
Let's talk about blood. It's the essence of life itself. But what happens when there isn't enough? Today on the Smart Biotech Scientist podcast, we meet a man who knows this terror firsthand. I'm David Brulman, and my guest survived a paragliding accident that should have killed him, saved only by an emergency blood transfusion that forever changed his path. Ari Gargir didn't just recover from his near-death experience, he's now pioneering technology that could solve one of healthcare's most desperate challenges: creating universal lab-grown blood cells for 100 million units we're short of every year. From his doctorate in human microbiology to leading roles at Thermo Fisher and Neuroderm, Ari's journey has been building to this moment. Now, as a founder and CEO of RedC Biotech, he's not just developing a product, he is creating a lifeline. Grab your coffee and notebook because in the next 10 minutes, you'll discover how personal trauma transformed into a mission that could save millions of lives worldwide. This is bioprocessing with a purpose, and I promise you won't want to miss a single word. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brulman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics at market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Ari, welcome to the Smart Biotech Scientist.
Hi, David. Thanks for inviting me. It's a pleasure.
Absolutely, it's a pleasure. Share something that you believe about bioprocess development that most people disagree with.
I think most people agree that it's difficult. Most people agree that it's unanticipated. Most people agree that you have to start as soon as possible. Most people will agree that you have to have a design freeze. Depends on your state. If you're a startup company, usually you want to start immediately. You want to say that proof of concept. You want to say that you have the capability already, and you want to start early. If you're a startup, you want to disagree with waiting until you have a fully designed process. That's the only thing I would think earlier companies would disagree with, and people that have experience will certainly disagree with. That's perhaps what I would take from this kind of a question.
Before we dive into this exciting topic you're working on, Ari, let's talk about yourself, because your journey from academia to entrepreneurship is fascinating. Can you walk us through that pivotal moment when you first fell in love with biotechnology and share some of the surprising or unconventional experiences that shaped your path now to founding your own company.
Actually, when I started learning biology, I had no idea what biotechnology is. I finished my military service and I knew I love biology. I love animals. I'm extremely interested in nature. And I had no idea about biotechnology. I started my bachelor's degree, and to me, most of biology was chasing butterflies with a net. I went to one specific course, which was Fundamentals of Biotechnology. And the professor that was teaching taught us about bioreactors. He was talking about these immense 120,000 liter vessels that are the size of a SpaceX rocket, and that you could grow things in it, and that you could grow materials that have great impact, like citric acid or penicillin. And that suddenly captivated my imagination. And I understood that that's what I want to do when I'll be a grown up. And from that point forward, I started focusing myself on biotechnology. More or less, when I was in my PhD studies, I understood that I do not want to be in academia all of my life, that the industry is what interests me for several reasons, for building a cooperative group, having a goal of developing an actual product that will have impact on people. When you're in academia, you may or you may not have impact. Could be interesting, it may not be interesting. But when you're in the industry, you have to make a product that will have impact, that will have commercial impact. So that's something that I understood that I want to do. And after a really short postdoc, I moved into my first industry role, and I led R&D for a very small startup. And from there, moved forward into several other startup companies and had a role in a couple of global companies. One of them was Life Technologies, and Invitrogen that turned into Thermo Fisher, and Mitsubishi Tanabe Pharma. In each one of these companies, I gained an experience that brought me to the understanding that I would also want to do my own company and bring all of those passions together. Big biotech, big bioreactors, making large-scale product, and having a great impact on the community, on humans. On the other hand, I also had another life-changing experience. When I was a bachelor student, I tried all kinds of things that had to do with parachute, either skydiving and paragliding, and I was terrible in each one of them. I hated skydiving, I hated leaving the airplane and parachute not opening automatically. And when I was doing paragliding, I crashed. And I had an accident. I had field surgery. I had lung injury. And I woke up in a helicopter getting a blood transfusion that saved my life. And I actually remember grabbing the doctor who did the field surgery and was accompanying me flying to the hospital. I asked him, it was back in 1990, HIV pandemic started breaking out. And I remember asking him, was this blood tested? And he said, yeah, it's okay, don't worry. And as I woke up again, it was already in the hospital. That sparkled an interest in the challenges of blood transfusion. And I understood that I have to find a solution for that. Many years later, I came across a technology originating from the Technion here in Israel that allows to grow stem cells, iPS, at large scale, and allows to produce these cells at a very low cost and differentiate them into your target. I understood that taking this together with differentiation of stem cells towards red blood cells, perhaps we can solve the challenges of red blood cell supply.
Wow, what a fascinating story. You experienced a life-altering event that now led you to founding your company and solve a big problem. So before diving into how this works and how you produce it, and you mentioned stem cells, so I'm really excited to talk about that. I think many of the people listening might not realize the scale of the global blood shortage crisis. Can you paint us a picture of how that looks like today and why you're solving such a big problem?
Red blood cells, as everybody knows, they're the oxygen carrying capacity of our body. The red blood cells are the ones who go into our lungs. They exchange oxygen for carbon dioxide, and they go to brains, to our tissue, and release oxygen, and in change take carbon dioxide from our body and release it in our lungs. Red blood cells are required in many medical interventions. There is not one hospital department that does not require red blood cell transfusions. If it's for trauma treatment, people lose blood in accidents or in other occasions, people going to surgery, women at birth. Something that not everybody knows, but about 200,000 women die every year from blood loss during childbirth, and many of newborns require blood transfusion because they're born anemic. So the mothers may lose a lot of blood and require blood transfusion to the newborns. And of course, there is a whole area of anemia. Anemia means lack of red blood cells. Anemia that is not caused because of trauma or surgery or loss of blood in a traumatic method. Anemia can be caused from hereditary diseases like sickle cell disease, thalassemia, nutritional problems, all kinds of diseases, liver diseases, all kinds of diseases that cause loss of red blood cells. Tropical diseases like malaria specifically hits red blood cells because the parasite lives inside the red blood cells. And cancer. In developed countries, about a quarter, 20% to a quarter of red blood cell transfusions go to cancer patients. That's an enormous number. So the use of red blood cells is required everywhere. Around the world, about 120 million blood transfusions are donated every year, but there's a calculated shortage, constant shortage of about 100 million blood units every year. So if you look at it, humanity is supplying maybe half of the blood that is really needed in order to save the lives of everyone or to maintain a life quality for everyone.
That's crazy. That means that we would need double of what we currently have, correct?
Right. If you look, about 60% of that blood is in high-income countries, which are still a fraction of humanity, which means that we have enormous numbers of people that suffer from blood inequity. And above all of this are the challenges, biological and logistical challenges of blood supply. First of all, you know, we have blood types. Distribution of blood types does not fit the actual need. We have the ABO groups, which are the prominent antigens on blood cells. We have the RHD, that's what gives the positive and negative. So let's take the O negative as a blood type that fits everybody. About 6% of the population are O negative. The actual need for O negative blood is more than double of that. So it's in constant shortage, meaning that the inventories of O negative are always short. The other blood types are in less challenge. Blood has a shelf life. It can be frozen if you add a lot of antifreeze to it, a lot of glycerol to it. And if you inject it then to someone, if you transfuse it, you have to get rid of it. But the shelf life of a blood unit is about a month. Its quality deteriorates after about 2 weeks. The shelf life is a problem. The biggest problem of blood supply is the donor. Okay, the donors are fabulous people. These are altruistic people, people that want to give and save lives, but they're not enough. And you know, even the good people, when there's bad weather, when there's an earthquake, when there's a pandemic, when there are holidays, they cannot come to donate, and there are shortages. So even in high-income countries where the donation agencies are working perfectly, they still have shortages because of the donors. In low-income countries, where the donation rates are mostly a quarter of what the actual need is, less than a quarter, so they are in constant shortage all the time.
So now, to put this into perspective, we have a serious problem. we lack about 50% of what we would need actually. And this is not just a 1-point event, it's constant. On top of that, we have pandemics, we have wars, we have accidents and so on that create additional peaks.
It will be worse, by the way.
Yes, getting worse, exactly.
Elderly population is growing. 75% of the blood goes people 65 years and higher. And the younger people are less happy to come to donate blood. So the shortages will be deeper and more frequent.
So tell us now how you and your company are solving this big challenge with your revolutionary technology.
So we're developing an industrial process to enable unlimited amounts of universal and donor-free red blood cells, both for transfusion and later on for advanced therapies. Advanced therapies meaning using the red blood cell as a platform for drug delivery. There are a few companies around the world that know how to load different therapeutics into a red blood cell and re-inject them and use them for different therapy areas. What we are utilizing is a technology that originates from the Technion. The Technion is Israel's MIT, and a specific technology that was born there in the lab of Michal Amit is a technology that allows to grow the stem cells as single cells in suspension, and of course in a chemically defined media, which means when you grow as single cells in suspension, it means that you can scale up indefinitely. Theoretically, of course, the engineering becomes the limit, but the biology is not the limit at that point. Of course, when you're producing using chemically defined media, it means that you could reduce the costs considerably. So as you grow in scale with the bioreactors, the cost of testing, the cost of harvesting, all costs, the economy of scale increases, and so the cost of raw materials. This technology allows us to scale up. We are using induced pluripotent stem cells, iPSCs for short, and these cells allow us to actually choose the biological characteristics of the stem cells that we work. And for this, we are developing our proprietary lines that will be O-negative blood cells. They'll be also have a more complex phenotype that will minimize the amount of side effects for a maximum size population. O-negative will save the lives of most people on Earth. I cannot say the exact number, but probably 95 to 98% of humans will be saved from an O-negative blood type. But there's still populations of people that are sensitive, have all kinds of immunological problems, or perhaps people that suffer from sickle cell disease and thalassemia are called heavily transfused people. These are people that get multiple transfusions every year. Could talk about people that get up to 50 transfusions a year, or that means almost a blood transfusion every week. So imagine that every week a person gets a biological sample from someone else. That rains havoc on your immune system. And these people are rejuvenated by the blood transfusion, but the feeling when the blood is first transfused is like getting the flu. And some of them have severe side effects. So by making a blood type that has the minimum side effects will enable to give these heavily transfused people a blood type that will minimize the effects. So by producing our red blood cells from a cell line that is derived from a universal donor in a bioreactor, make sure that, first of all, the blood will have no pathogens. It will have no residual blood components that cause all of these side effects. The blood will be much more reproducible. Donated blood depends on the individual that donated the blood. It depends on the lifestyle that the donor has, his nutrition, how much sport he does. The variability and the reproducibility of the blood and the quality all together will be much higher.
Yeah, absolutely, because you will be able to control. It will be a controlled process, and you will exclude the natural variability with your process.
Right. And most important, donor-free. The production will be according to forecast, according to demand. And it could be planned so that you will have minimum shortages on one side. And also, if you need to plan emergency inventories into it, you could plan it and use it, etc.
And that's where we'll pause for today in our fascinating conversation with Ari Gargir. We've heard how a life-threatening accident sparked a vision to transform global blood supply systems and the staggering scale of the problem RedC Biotech is addressing. Join us next time as we dive deeper into the bioprocessing innovations behind creating universal blood cells and the challenges of scaling this groundbreaking technology. Before we go, are you feeling overwhelmed by the complexity of your own bioprocess development? Book a free consultation to refine your CMC development strategy and propel your success. Remember, science might give you headaches, but biologics drug development should not. I'm David Brulman, and this is the Smart Biotech Scientist. See you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.